Hello everyone and welcome back to Bio 181 Lab. Today we'll be discussing mitosis and meiosis. So originally we all start out as a single cell, and I want you to think about the fact that we go from a single cell to the average human body containing about 37 trillion cells. Now think of yourself 7 years ago. Today you are a new set of cells compared to back then. In a certain way this makes sense. We know that our cells are getting old and dying all the time. If you've ever experienced dry flaky skin, that's old dead skin cells sloughing off. But your body just can't let cells die because eventually you'd have fewer and fewer cells in your body until you were nothing. Your body has to replace them. So how does it do that? It does that by cloning them. The cells make identical copies of themselves, or near identical, with maybe a few mutations per replication. And one cell becomes two. And this is a picture of that. So can anyone tell me how many chromosomes worth of DNA a human body cell has? So the correct answer is 23 pairs of chromosomes, so a total of 46 chromosomes. You may have heard of the company called 23andMe, which does human genome sequencing. So mitosis and meiosis is one of the more difficult lab concepts to understand, but if you start out with a solid understanding of the definitions in meiosis and mitosis, it's really not that difficult to grasp these concepts. So we're going to cover some of the basic definitions before we get into the mechanisms and processes of it. Let's begin with chromosomes. Chromosomes are the cellular structure where genes are located for the cell. They contain DNA, but also have proteins that are important for a variety of activities and reactions, as well as maintenance of the DNA. Some organisms have many different chromosomes than just one. For example, humans have 23 different chromosomes, and each one of those chromosomes is made up of a homologous pair, or a pair of chromosomes that are similar, kind of like having two versions of the same book. So in the human library or the human bookshelf, you would find 23 different books and two different versions of each one of those books on the shelf. One copy or one version of the book from the father and one version from the mother. And each version will differ slightly from each other, making for a total of 46 books on the shelf. So now that we've discussed what chromosomes are and what homologous pairs are, let's further our discussion about ploidy. Ploidy refers to the number of each set of homologous chromosomes. So for humans, we discussed that there's two sets of each chromosome. So there are two versions of each book. And so for human cells that have a full bookshelf of two copies of each chromosome, they're referred to as being diploid or diploidy, meaning they have the two copies of each chromosome. However, there are some human cells that only have a single copy of each chromosome. So in that cell, if you were to zoom in on their chromosomes, you could only count 23 chromosomes per cell. And because a normal full set is diploid, they refer to a human cell that only has a single set as haploid, which means half, or a single set of chromosomes. And in the context of other organisms, organisms can have any number of homologous chromosome sets. They can be triploid, tetraploid, hexaploid, etc., etc. And this is a visual representation of what that would look like. So here we have in the haploid cell, here we can see that there are 3 different types of chromosomes. And this organism is a diploid. So if haploid's the half copy number, diploid is 2n. And we see that there are 2 copies of each one of these chromosomes. So these are each homologous pairs to each other. So now we've discussed chromosomes, homologous pairs, and haploid or diploid. So this is where things get a little bit more confusing for people because they tend to confuse a homologous pair with what a sister chromatid is. So imagine when this diploid organism, just like our typical human cells, replicates its DNA, it has to make 2 copies of all of those chromosomes, all 46, right? So in order to do so, it makes a full copy of each book. There's a pair of, there are 2 homologous chromosomes per chromosome, and each one of those has to be made a copy of. So if you look at this image here, here is our diploid set of homologous chromosomes. Okay, so 1 copy of the book and another copy, another version of the same book. And in order for that cell to make 2 cells, it has to write down a copy of each one of these books. So it goes through a replication process. And now we see that version 1 of this book and version 2 of this book have been replicated. Now, if I was to ask you, is which one of these sets are diploid? The answer is they're both diploid. They both have 2 copies, 2 versions of the same book. It's just that this one now has an extra copy of each version. So when you look at these X's here, these X's are a cartoon depiction of what we call sister chromatids. Sister chromatids are when you've made a second nearly identical copy of one version of one chromosome. So we have 2 chromosomes here. Let's say this one's from the mother and this one's from the father, right? And they're homologous chromosomes. And now we've made an extra copy of the mother's chromosome and the father's chromosome. But there's still only 2 homologous chromosomes here, making this a diploid cell. The only thing that's changed is we've made an extra copy of each book so the cell can replicate into 2 cells. The process of creating 2 cells from one cell is a little bit more complicated than that. It's made up of many phases here, and this process is called mitosis. So we can zoom in on this process of mitosis and break it down into steps, because it doesn't just happen like that and you've got a new nucleus. There is a set of very well characterized steps that take place that lead to the replication of the nucleus and the partitioning of the proper chromosomes to each nucleus. Those steps are prophase, prophase, prometaphase, metaphase, anaphase, and telophase. On this diagram, you also see interphase at the beginning, which is where the individual cell is in its normal growth phase. And at the end, you see cytokinesis. Cytokinesis is where you create a new cell. Cyto meaning cell and kinesis meaning to create. So at the end, it's where the cell actually divides after it's completed mitosis. So the 5 stages in between are the stages of mitosis. So let's begin before mitosis. There are 2 copies of DNA in the nucleus. However, chromosomes have not yet formed, and instead the DNA in the nucleus exists in its unwound form called chromatin. So we can see here and here, right? In prophase, DNA begins to condense into chromosomes. Remember, we said that there was also proteins there, and it's a specific type of structure that occurs. So the nuclear envelope begins to break down. So usually there's a membrane that surrounds this DNA that kind of keeps it all in place. And then there's something called spindle fibers, which are these fibers that form and stretch across the cell. Next is prometaphase. And in this step, proteins attach to the centromeres, which are the middle point of that X, and they form something called the kinetochore. And this kinetochore is where the spindle fibers attach to the chromosomes. And once the spindle fibers attach to the chromosomes, they begin to pull the chromosomes to move along the equator of the cell. So we're going to see in the next slide how that looks. So prometaphase. And then now we actually have a full metaphase here. And so the spindle fibers help in aligning the chromosomes in a straight line at the center of the cell. This line is called the metaphase plate. And such an alignment helps in proper separation of chromosomes and ensures that each daughter cell gets one copy of the chromosome. The next step is anaphase. In this phase, the sister chromatids separate at the kinetochore. Remember, the sister chromatids are almost perfect copies of each version of each chromosome. So each homologous pair is replicated into a sister chromatid. And as you can see here, those X's get split apart. So now we just have a single tail for each single copy of those homologous chromosomes. So proteins composing the centromere of the chromosome are degraded, and the sister chromatids are pulled apart to opposite ends of the cell by the spindle fibers. So remember, for example, with humans, we have 26 chromosomes, and each one of them have homologous pairs. So there's actually 46 total chromosomes if you were to count them up. When sister chromatids are made, each one of those 46 chromosomes, copies are made of them. And they're still stuck to their original copy, and they look like an X. And in this anaphase process, they are ripped from their exact copy into two separate chromosomes. So here we see kind of like these two books are being pulled apart from one another. And so on each side here, you're going to find that you have 46 chromosomes, a homologous pair of each of the 26. And now we're at telophase. At this point, the chromosomes reach the opposite ends of the cell and expand. The formation of that nuclear membrane also begins around these chromosomes. And finally, the spindle fibers disappear and cytokinesis begins. Remember, cytokinesis is the procedure in which the two daughter cells separate. So a fiber ring made up of actin, which is a type of protein, forms around the center of the cell and pinches the cell to make it split into two diploid with 46 chromosomes altogether, daughter cells. Again, diploid for human cells describes cells that have a full set of chromosomes. And haploid describes cells that have a half set of chromosomes, or a single copy of each chromosome. So, why are haploid cells around and what are their importance? Well, they play a role in meiosis, which is the process of sexual reproduction. A way for you to memorize this is it has a letter E in it. So just like sex has a letter E, so does meiosis. And so Cells that are haploid in our body are our gametes. So in humans, they're going to be the sperm and the ova, or the egg. And the reason for this goes back to the set of chromosomes and where they come from. If you remember, each pair of homologous chromosomes, you get one from the father and one from the mother. And so if you wanted to make a set of 46 and it had to come from two cells fusing together, then you would have to have one cell that only had 23 and the other cell that only had 23. So when you combine their two bookshelves, then you get the 46 books. So because the gametes need half the number of chromosomes, there is a special process for making them, which differs from the process the rest of your body's cells sees. That is just plain mitosis. We've already mentioned the name of that process. It's called meiosis. Meiosis is a lot like mitosis, and its phases even go by the same names of prophase, metaphase, anaphase, and telophase. But in meiosis, the division process happens twice. So you have what's called meiosis 1, which we see here, and meiosis 2. There are a number of similarities and a number of differences between meiosis and mitosis across their sets of events. And as you simulate this process in the lab, it'll be your job to identify these similarities and differences. In this slide, we start from the same cell. We have a 2N cell that already has its chromosomes replicated. So it's already formed its sister chromatids. And on one side of the slide, we have mitosis, which again only goes through one set of DNA replication and cytokinesis. And in the other side of meiosis, we go through meiosis 1, and then we go through meiosis 2. So you can see, again, it has prophase, metaphase, anaphase, telophase, and then another prophase, metaphase, anaphase, telophase. So again, as a review for mitosis, in prophase, we have our sister chromatids that get attached to the kinetochore and the spindle fibers. These cause it to align in the metaphase plate. Those sister chromatids get split apart here and get pulled to the opposite ends in telophase, and then a nuclear membrane is reformed around them. And then we have cytokinesis take place, and then we have interphase, which is the normal cellular growth part of cells. So meiosis starts off the same, but its first phase is a little bit different. See, in metaphase, in mitosis, we saw that the sister chromatids line up. And then in anaphase, each chromatid is separate. And so here we have the formation of a single set of this diploid organism. It has two different chromosomes and two homologous ones of each one that end up in the final cell. So here we see 2N. So that means it's diploid. A haploid cell would just be N. So we've started off with the same sort of cell in prophase in our meiosis. We have made our copies of each pair. So we have two sister chromatids here, two sister chromatids here. What happens is so here's a mother and here's the father copy of these chromosomes. They end up lining up next to each other, and they go through a process in which they exchange genetic information, which we'll see. So first of all, in metaphase 1, we have our sister chromatids kind of line up along this metaphase plate. But the big difference is in anaphase 1. In anaphase 1, if you'll notice, the sister chromatids aren't split apart. Rather, the homologous pairs are split apart. So mother and father's copies are put into separate sides of the cell. So now we've made a haploid set, two haploid cells. Here we see in prophase 2, we have two haploid cells. They might have a copy of each homologous chromosome, but they only have one of the set of homologous chromosomes. So if you compare prophase here to the prophase in its neighboring cell in mitosis, you'll see in mitosis it has two homologous sets, whereas there are no homologous sets in this new cell. It's n equals 1. So this is how we form our haploid cells. Now in this meiosis 2 process is just separating those sister chromatids. And so we actually go from one cell to two cells, and then in order to make a single copy of those chromosomes, we have to again make them into two new cells again. So this haploid cell again goes through metaphase where the sister chromatids align, and it's just like you see in metaphase in mitosis. And then in anaphase, we have a splitting of those sister chromatids. And so they get pulled into new sections of the cell. And then you go through telophase and cytokinesis. From the start of meiosis 2 to the end of meiosis 2, they are haploid. In mitosis, the start of mitosis to the end, they're always diploid. Never do these cells not have two copies of each chromosome. Okay, so in meiosis 1, they remain diploid for the cell, and in meiosis 2, they are haploid throughout the cell. My apologies. Actually, in telophase at the end of meiosis is when they become haploid as the cell splits. So anyway, those are the major differences between the two. And in this very first prophase 1, I would like to review in meiosis the two copies of the sister chromatids, one from the mother and one from the father. They actually have this process called crossing over, and that's where if you were to take, two versions of the same book, what you would end up doing is cutting out some pages from one version and then splicing them into the other book for the exchange for the other book's pages to put in the previous book. So you get kind of a hybrid of each book. Neither book is the same version anymore. It's a mix of both versions. And that's kind of the beauty in sexual reproduction, is by mixing up the DNA on each of these chromosomes, you get a lot more diversity. So now I have some examples of some sane cells with the different phases of the cells. So if we take a look here we have interphase, where the cell has its DNA, it's not in its condensed form, it's within a nucleus here, and this is the part of the normal cell cycle. And now here we have prophase, where the DNA is starting to condense into those chromosomal structures. In metaphase, we see that they are aligning along that metaphase plate. And then in mitosis, those sister chromatids get pulled apart. So we go from having kind of these X's into just these straight ones where the sister chromatids are yanked apart. And then in telophase, you really see that DNA being condensed again, or the structure of the chromatid is being degraded, and the cellular envelopes are starting to form around the cell. And this image right here just shows the difference between metaphase and prometaphase. And here's another example of a prophase where you don't quite have this metaphase plate being formed yet. And in the prometaphase, they're still being pulled by the spindle fibers to be aligned along this plate. So something that you should be able to say are, if this chicken is diploid in its normal cells, are its eggs diploid or haploid? I'm actually going to leave you up to figure that out yourselves. Thank you very much for joining me. I hope this was helpful, and I'll see you on the next round.